Hello, everyone. I am super thrilled to be moderating today's Grand Rounds because this is a topic that has always thrilled and excited me, which is how our gut microbiome influences our immune health. Additionally, this is such an amazing example of what UCSF brings to the research, which is this idea of bringing bench science to the bedside to patients, enrolling patients in clinical trials, and really watching the discoveries that happen in the lab transform the lives of our patients. So you will hear about that from two speakers today, who will be talking about how the gut microbiome and the role that plays in rheumatic diseases like rheumatoid arthritis. Our speakers will highlight the impact of the gut microbiome on therapeutic efficacy and how gut bacteria metabolize drugs and influence the body's autoimmune processes. Our two speakers today, I'm delighted to welcome them both. Our first speaker will be Jonathan Graf. He is a professor of medicine and works in translational research, this idea of bringing bench discoveries to the bedside for our patients. And he co-founded the UCSF Rheumatoid Arthritis Observational Cohort and is the director of CSTARE, the Center for the Study of Advanced Immunotherapies in Rheumatic Diseases. This center specializes in early phase clinical trials and studying novel therapies and the impact on the disease process for our patients. Next, we'll have the privilege of hearing from Dr. Renuka Nayak. She is an MD, PhD, and a physician scientist dedicated to advancing the care and treatment of patients with rheumatologic conditions, such as rheumatoid arthritis, lupus, psoriatic arthritis, and other autoimmune diseases. She has a background in both biology, computer science, and clinical rheumatology, and uses this unique background to investigate the role of the human gut microbiome in the treatment of rheumatoid arthritis. You'll hear her innovative approach to using patient specimens and microbiology and the next generation sequencing to better understand how our microbiome really influences disease for our patients. So I'm delighted to welcome Dr. Graf to the podium first.
Thanks so much, Michelle. It's a pleasure to come speak here today. Before I turn it over to the microbiome part of the talk, I thought I would give a little overview very quickly, since my time is pressed and quite limited, on RA and sort of some novel advances that have occurred just in the last few years that provide insight to potential novel therapeutic approaches that'll be coming, hopefully, to a clinic near you. And so with that, let's see if this will advance. Seem to be, there we go. So these are my disclosures. I do a lot of work, obviously, in early phase clinical trial development in RA. Rheumatoid arthritis is a systemic disease whose predominant manifestation involves a chronic inflammatory small joint polyarthritis. But it's a systemic disease. So the disease isn't limited to just the joints, but as you can see in the visuals over here, can affect other organs throughout the body as well. The disease is particularly chronic and progressive, and certainly in the joints, this leads to permanent joint damage, disability, but also an increased mortality, probably from the chronic burden of inflammation that patients suffer from over the course of their lifetime. It's quite prevalent. Up to 1% of the US population is afflicted with this disease. It has a curious predilection for females more than males, 3-to-1, which is something that is an interest of many people in research. And it's a disease where the peak incidence is usually in young women in their 30s and their 40s. And so given that this is a chronic disease, people are living with this for decades at a time. And that's a point I'm going to make over and over again during the course of this talk. Of course, the disease can have its onset at any age, but most patients are living with this chronically throughout the entire course of their lifespan. Now, the way we approach treating rheumatoid arthritis in this era is a treat to target kind of approach. It's not a novel approach in terms of other chronic diseases. We do this for high blood pressure. We do this for diabetes, for example, where we target hemoglobin A1C. And RA is really no different. We have objective outcome measures that we use, disease activity scores, for example, that help us gauge how a patient is doing at any given cross-sectional point in time. But we start patients on a conventional synthetic DMARD, disease-modifying anti-rheumatic drug, like methotrexate. And then we bring them back to clinic, assess how good their response is in an objective manner. And if a patient has not achieved a low disease activity target, we then will add additional DMARDs. Often these are biological DMARDs, like you may have heard of anti-TNF therapies, for example, or we can use a combination of conventional synthetic DMARDs. And we do this sort of in a trial and error approach. Putting patients on therapies, adjusting doses, bringing them back, seeing if they're in a low disease activity state or not, tinkering with therapy even further, adding additional drugs. And this can go on over the course of many, many years of a patient's life. And if the disease continues to remain active, we continue to ascend this pyramid, trying or adding other therapies to a particular patient's regimen. And as my son here will attest to, he loves climbing walls. He loves to climb up walls as high as he possibly can. And in RA, we really don't try to do this. We think that we have this tremendous armamentarium of biological therapies that have come into the marketplace over the last couple of decades. But the reality is, again, this is a chronic disease that patients are living with over the course of their lifespan. And many patients actually have to keep ascending the pyramid over the course of their lives, get to the top of the pyramid, and still have not achieved the type of disease control that we would be otherwise satisfied with. And this is highlighted by a very, very nice study now published about 1.5 years ago in Science Reports, looking at a cohort of real world. This is not a clinical trial, but this is a real world cohort in France. And it actually follows patients over 7 years. And you'll notice, as I put on the bottom here, during the emergence, not only of the treat to target therapy strategy, but also the emergence of additional classes of biological agents that have come to market. And if you look here at the end, see if I can show this here, these patients over here, at the beginning of the study, about 30 patients were in remission. And by the end of this real world cohort, 7 years, because of the advent of all these different drugs you can see here, abatacept, rituximab, tocilizumab, and eventually the JAK inhibitors, we've been able to achieve remission in about 60% of patients. So that's a significant improvement, right? But the glass may be half full on one end, but think about the half full glass on the other end. Despite all of these biological therapies that we can use climbing the pyramid, more than 40% of patients are still not in remission. And therefore have this excessive inflammatory burden and symptoms that they're living with over the course of their life. Another real world example of this is to look at our holy grail therapies, or so we think, the anti-TNF drugs. When these drugs came to market, I don't want to belittle these revolutionized the care for our patients with RA who previously never could imagine achieving the types of results we see with these drugs. And in fact, this is another real world study, but if you look in the real world, when patients start an anti-TNF therapy, about 70 to 80% of them get a 20% improvement in their joint scores, which mirrors very closely what was seen in the clinical trials. So we pat ourselves on the back and say, wow, these drugs are fantastic, right? But you have to remember something. That's a 20% improvement overall that we're measuring here, right? The bar is not the highest bar. And in fact, if you look 8 years out at patients on adalimumab, for example, which is Humira, an anti-TNF drug, only 38% of them are still taking the drug, meaning that the majority of those patients either weren't adequately controlled initially or eventually lost their control under this drug and had to switch and again started cycling through medicines, climbing that pyramid that I showed you earlier as well. One last study that I'll sort of highlight that sort of points this out. Even when we control the disease really, really well, okay, this is a study that looks at patients on anti-TNF therapy who are in remission and have been in a durable remission for more than 12 months. So these are the patients we are thrilled with. These are the glowing successes in RA therapy. This is a trial that said, how long do we need to leave these people on therapy? Do we need to leave them on their drugs for 40 years? And so it was a study that looked at tapering their anti-TNF therapy. And when you do that, compared to a control group that does not taper their therapy, and this was a nice randomized controlled trial, 65% of patients within 12 months flared, as opposed to almost no one who continued on their anti-TNF therapy. And so what that tells us is that even in patients in whom we have in remission, whom we're controlling the disease beautifully, right, the fire is still smoldering under the surface. We haven't done a great job in altering the underlying autoimmunity that's driving the disease. We're temporizing the inflammation on the surface. And as soon as we, release the lock, so for example, on the door swings open and the inflammation comes out and the disease begins to flare as well. And so again, it tells us that as good as our drugs are right now, they aren't doing anything to change the underlying balance, immune imbalance that occurs in our patients as well. So even in an era of multiple biological therapies, a sizable number of patients fail to reach remission for their disease activity. Many patients who at first respond to treatment lose their response over time and thus end up cycling through treatments. Patients who are otherwise, like I just showed you, in a long-term durable remission on their current biological therapies will flare if you happen to stop the therapy, which suggests, like I said, that the immunologic that the current immunologic therapies do not restore immunologic homeostasis. Now, this was recognized by Francis Collins, who was the former chief of the NIH. And what I think of his lasting legacies before he left was to launch something in 2014 called the AMP, the Accelerating Medicines Partnership. This was a unique public-private partnership between the NIH, the FDA, biopharmaceutical companies, I think 10 biopharmaceutical companies, and nonprofit research organizations like UCSF, which is a member of the AMP, particularly in the lupus side of things. And the goal was to transform the current model for developing new diagnostics and treatments, right? This problem was recognized. And the way that Collins envisioned this would be done through an improved understanding of therapeutically relevant biological pathways, and that he would use validated techniques that would leverage big information to help and novel technologies to help in the development of potentially novel therapeutics for various diseases. And this is not limited to autoimmune diseases. The initial seed money was about $230 million in 2014, and it funded a bunch of 3 to 5 year pilot projects. And again, not just for autoimmunity, which of course for us was RA and lupus, but also included research in Alzheimer's and type 2 diabetes as well. And again, this is a diagram that Collins used when he was sort of shopping this proposal around. Current drug therapies are being developed using animal models and traditional cell lines and things like that. But the way the AMP was functioning is it really was going to leverage, like I said, emerging technologies, DNA and RNA, single cell sequencing, proteomics, bioengineering cells, imaging, as well as big data, demographic, clinical information, healthcare records. Tissue and blood samples were a very, very important part of this, and using it to identify the biological pathways that were relevant for those various diseases. And just now, we're beginning to see some of the fruits for those sort of seed investments that were made 10 years ago as well. I don't know if this particular research was a byproduct of the AMP, but this lab, Mike Brenner and Deepak Rao's lab at the Brigham, is absolutely a major center for the AMP. As well. And so there's no doubt that funding has flowed down to do this kind of work. This is fascinating stuff. So about 6 or 7 years ago, a new subset of cells was discovered that appeared to be pathologically expanded in patients who had RA. And this was done, again, using sort of next generation sequencing techniques they seek, using actual patient samples. And what you can see here on the right is in a normal lymphoid tissue like a tonsil, there are T follicular helper cells. These cells express high levels of PD-1, which we'll talk about in a second, and the ligand CXCR5 that hones those cells to the lymphoid tissue. But in RA patients, there was an expanded population of cells of T helper cells that were also PD-1 high, but did not express CXCR5. So they don't hone to lymph nodes where they belong. So they look for all the world like T follicular helper cells, but they're in the periphery. And they're actually in inflamed tissues, in the synovium and in the synovial fluid of patients with active inflammation. And so these were called T peripheral helper cells. And so the question was, what are these folks doing? They're conspicuously there, and they must be doing something. And it turns out that these cells do lots of things, but the crux of what they're doing is secreting cytokines and chemokines like IL-21 and CX. Responsible for B cell maturation and differentiation into plasma cells. It's involved in germinal center formation and making those B cells, whipping them to make immunoglobulins. So B cells are very, very important in RA pathogenesis. They also secrete this cytokine, I said, CXCL13, which is critical to get B cells to migrate to the sites of inflammation, right? So these T peripheral helper cells are not acting very well. They're helping B cells do some very, very bad things in the joints of patients with RA. And so T peripheral helper cells are very conspicuously present at tissue sites of pathology and not in the lymph nodes where they should be. They provide help to memory B cells at the site of inflammation, and they tell the B cells, hey, you need to start making antibodies and doing other things that potentially are driving the pathogenesis of this disease. And it therefore suggests there's a potential benefit to targeting these particular cells and maybe this PD-1 pathway that I'll show you in a second to treat patients with RA. So stay tuned for that in just a couple of minutes. Another byproduct of this sort of medical partnership and collaborative effort that I told you about was work done by Dana Orange at Cornell. This was published in the New England Journal a few years ago. And Dana's developed a very novel way to study a chronic disease. So it's very hard to do a clinical study or a clinical trial when you're seeing a patient once every month or every 2 months, right? They're flaring when you're not seeing them. And so she's come up with a way to send patients home with these little finger stick kits and collect little capillary tubes of blood on a weekly basis, like you can see over here at home. And she intermixes these home collections with clinical visits and clinic collections as well, and has patients reporting their own disease activity while they're at home in sort of a diary sort of way. And so she's able to capture patients, you can see here in the purple, when they flare, not only when they flare, but also in the weeks antecedent to those flares as well. And again, she's not collecting enough blood to look at the cells themselves, but she can begin to look at the RNA transcriptomes in the blood prior to patients actually flaring. And what she found is very, very interesting. At the actual time of the flare, if you look here on the top, right, when patients are flaring, there's a gene signature here that's red hot. And it looks for all the world like neutrophils and monocytes and the types of cells you see in an acute inflammatory process. So that's a no-brainer. But if you look upstream, 2 weeks upstream, there's this cluster 2 of cells where the RNA map is very, very hot. And this resembles the transcriptome of naive B cells. So B cells are being activated and are starting to circle around 2 weeks before people are flaring. And then 1 week before people are flaring, there's this, what was previously felt to be an unknown cell type that for all the world, if you look at their transcriptome, see if I can find the pointer here. If you look at their transcriptome, for all the world, looks like fibroblasts. So fibroblasts are supposed to be in tissues, right? Laying down collagen and stuff, lung, joints, things like that. But these are circulating fibroblasts that appear to very accurately presage the development of inflammation and flares in RA. And so she called these prime cells or pre-inflammatory mesenchymal cells of who knows what origin. And so this was a very, very surprising discovery. Why this is important is, as first I showed you, that dense sampling is a technique which we can use in other areas as well to study chronic and relapsing inflammatory diseases. It's going to change the way I think we're able to study these diseases in real time. But I think this is also important because even in a chronic inflammatory autoimmune disease like RA, okay, where patients have had disease for decades, maybe even more, right? Patients have had a preclinical phase and stuff where you'd figure their immune system is already activated, T cells are already activated, they've already seen their antigen, the horse is out of the barn, now all you can do now is just control the inflammation, right? Well, this suggests, since you can see naive B cells in the bloodstream before patients flare, it suggests that there's a pool of naive B cells and T cells, maybe those T peripheral helper cells I just spoke about, that are capable of initiating and perpetuating disease flares all over again, right? And so this supports the potential therapeutic benefit to targeting the upstream pathways in autoimmunity, not just the end inflammatory pathways as well that we have available to us today. And in particular, there's a benefit to targeting B cells, I think, in this disease. Now, we've known for a long time that B cells are not innocent bystanders in rheumatic diseases like RA. They can certainly promote a lot of pathology. They can serve as antigen-presenting cells. They can produce pro-inflammatory cytokines. And as we all know, and you can see over here, they can generate autoantibodies, which can form immune complexes and deposit in tissues like the lung and the joints and places like that, and induce joint destruction, inflammation, complement activation, and all that other bad stuff as well. And so we've known that you could target B cells as a therapeutic target in RA. This is the New England Journal study in 2004 that led to the approval of rituximab to treat RA. And when the rituximab came out, and we use plenty of rituximab today to treat RA, you could see that the results were fairly impressive. These were very, very refractory patients who got roughly a 70% improvement in their RA. A 20% improvement was achieved in about 70% of patients. And so, we were patting ourselves on the back. Look, B cell driven therapies work really well. Well, again, if you're a half empty, glass empty type of person, you might argue, well, what about the ACR 50 and the ACR 70, the 50% improvement and 70% improvement? And as good as rituximab is at depleting B cells, only about 30 to 40% of patients get a half improvement in their disease. And even fewer, about 10 to 15%, see a 70% improvement in their RA. So rituximab is hardly a panacea. And one of the reasons for this very well might be that if you look in the periphery, you can't find any evidence of B cells existing immediately after a patient's been treated with rituximab. But if you look in the tissues, and in particular, this is looking in lymphoid tissue as well. Here are patients on the top pre-treatment with rituximab, and these are patients post-treatment with rituximab here. And although it's a little bit blurry, what you can see is that the brown staining in here doesn't appear to change very much. And that's because rituximab is not depleting B cells from the lymphoid tissue itself. In fact, many of these germinal centers are structurally still intact where the B cells are residing as well. So rituximab is probably addressing the right target. It's just not doing a good enough job. And so what do we have coming on the therapeutic horizon right now? Well, there are molecules called BiTEs. And for those of you in the hematology oncology world, you're very familiar with BiTEs because they're used to treat various liquid tumors right now. BiTE is short for the term bispecific T-cell engager. And this is an antibody-derived dual-specific molecule that can bind to both B cells and T cells. And so it's a matchmaker. It's a typical matchmaker. It brings B cells and T cells together, only this isn't for a wedding. This is for a funeral, because the job of these T cells is to kill the B cells, which is exactly what they do. So B cells will apoptose and die when in the presence of these T cells brought closer together to them. There are a lot of advantages to this. This is a very effective way to kill B cells, and these molecules can penetrate tissues probably a lot better than drugs like rituximab can. And if you look at these are clinical data published just a couple of months ago in Nature Medicine. If you look on the left over here, you'll see that after the BiTEs are given, within a few days, you see this heat map light up of inflammatory genes. And that's to be expected, right? The T cells are engaging B cells. They're releasing a lot of cytokines. They're very angry. And you see this inflammatory process that occurs, which very quickly calms down. And on the second cycle that patients receive with BiTEs, you don't see that inflammatory signature lighting up anymore because the B cells have been depleted. And in fact, if you look out a few more days beyond that, the heat map goes cold completely, right? The genetic heat map. And this reflects exactly what we see what was seen, I should say, in the 6 patients who received this drug. This is only 6 patients who've gotten this so far. And what you see these are, I can tell you, extremely refractory, extremely severe, the worst of the RA patients you'll see out there as well. And in fact, all of them achieve, go down in terms of the tender and swollen joint counts, go into practical remission. And in fact, if you look at a formal disease activity measure in these patients, the cutoff for remission is this disease activity score of 2.6. Just about everybody goes into remission. Okay, we're not talking ACR 20s here or 50s or 70s. We're talking remission for the most severest of patients who get this very intense B cell therapy. And again, if you look at the tissue, unlike what I showed you with rituximab, this is now looking in the synovial tissue itself at both the presence of B cells and plasmablasts before and after treatment with these BiTEs. You can see that the B cells are almost completely obliterated as well. So again, this is a good opportunity to sort of summarize that therapies that more completely target and deplete B cells may have improved efficacy in treating RA, especially those with the most refractory disease. And this also raises questions for what about CAR T therapy? We have outstanding CAR T products that are out there that will also obliterate B cells from patients as well. But CAR T therapies may come at greater cost. They have a side effect profile, including cytokine release syndrome and neurologic side effects as well. There's a risk of lymphomas. There's a lot of cost and delay that go into manufacturing these products because they're autologously derived. There's a need to condition patients with chemotherapy before they receive their own autologous grafts back, and they have to be hospitalized for a while too. So these BiTEs may not be quite as effective maybe as potential CAR T therapy is going to be, but they potentially come with a lot better side effect profile and potentially easier to use as well. So with that, let's go back to PD-1 for the last 5 minutes of my talk here. PD-1 is an essential component of what we call immune checkpoints. There are several checkpoints in the immune system, but when a T cell is activated, it sees its cognate antigen, and in the setting of an appropriate co-stimulatory stimulus, T cells will activate. But there comes a time when all activation must subside, right? My kids love running around all day, but at 8 o'clock at night, it's time to stop, tearing the house apart. It's time to go to bed. And the same is true for T cells as well. And so when they become activated, they upregulate a molecule called PD-1, which binds to a ligand on the antigen-presenting cells and energizes these cells. Actually, it energizes, but downregulates these cells to sort of put them to sleep, tell them to stop being activated as well. It's a very effective signal. And in fact, cancer cells have hijacked this pathway to sort of put anti-tumor T cells to sleep and allow the tumors to actually grow. And blocking these pathways with antagonists have been very, very effective therapeutic products in oncology, for example. They've revolutionized the care of lung cancer, melanomas, bladder cancers, all that kind of sort as well. Well, the opposite is true. If you can antagonize this pathway to great effect, what about agonizing this pathway with antibodies? And sure enough, there was a study last year in the New England Journal of a drug called parasolimab that is an agonist of PD-1, right? A drug that will turn off the activation signal in T cells. And this was looked at in patients who were refractory to standard RA therapy. And you can see there was a significant improvement in disease activity in patients who received parasolimab. Now, I can tell you hot off the press from two or 3 days ago, the company that makes this drug killed the program. I mean, it has a lot of other drugs that it's making to focus its efforts on, but I can also tell you that it's probably not the best agonist out there. There are better agonists, including ones that we're working on here at UCSF. And these agonists not only can bind to PD-1, in particular the PD-1 on those T peripheral helper cells, the bad actors I told you about, and they can agonize these cells, but they can also bind to PD-1. And because of the nature of these particular types of antibodies, they can deplete them as well. So they're both agonizing and they're depleting. And because they're depleting, this has the potential to actually restore immunologic balance, immunologic homeostasis in patients who get this therapy. And so I'll show you what that looks like. If you look at patients who receive rosnilimab, which is the drug we're studying here at UCSF in partnership, you can see that patients who that their response to tetanus is actually reduced when they receive this drug at increasing concentration. So the T cells, the recall response to tetanus, can be agonized alone with this particular antibody compared to a very sophisticated control. But in addition to the agonist effect, if you look at those cells in study subjects who are the highest expressors of PD-1, those T peripheral helper cells, for example, they're depleted post-treatment. In cells that are intermediately expressing PD-1, they're reduced, some modest amount. But most importantly, in the PD-1 negative cells, this is our T cell repertoire that's resting. It's waiting for the next infection at some point, right? This doesn't deplete them at all. So the immune repertoire is completely intact. It's like a selective, selective circumcision, so to speak, of these pathogenic T cells out of the T cell repertoire, leaving the rest of the T cells completely intact. And so in essence, if you sort of look at this schematically, we can take someone's immune system that maybe is skewed towards an autoimmune phenotype with these PD-1 high activated T cells, selectively deplete them and restore immunologic balance, where the T cell repertoire looks more representative of someone who doesn't have RA. Okay, and that's the promise of these drugs, not just treating inflammation alone, but maybe restoring immunologic balance. And so in the last 30 seconds, I'll talk about a final pathway that looks at restoring immune balance to the immune system. So this is looking at T regulatory cells. There are different kinds of T cells in our body. There are T effector cells that activate the immune system. And then there are regulatory T cells that provide counterbalance, right? That also help, it's not just PD-1 that we rely on to control T cell activation. We have other T helpers, T regulatory T cells that will help suppress activated T cell, effector T cell activity as well. And this prevents overactivation of the immune system, which includes autoimmune diseases and allergy, for example. Now, like effector T cells, regulatory T cells are also antigen specific. But when they see their antigen, they can exert their regulatory effects sort of in the general neighborhood around them as well to sort of control inflammation. And so just like we have effector CAR T therapies being used in cancer, there are companies, including one that was founded by Jeff Bluestone, who's the former provost here at UCSF, where he can take regulatory T cells and transform them, basically transfect them with a CAR construct that is antigen specific. So he can make antigen specific T regulatory cells that recognize the autoantigens in rheumatoid arthritis. And we now know through a lot of research that the autoimmunity in RA is directed against a post-translationally modified proteins that contain citrulline. It's one of the reasons why anti-citrulinated protein antibodies are so specific for RA. And the idea is he can engineer these cells. They're autologously derived. They don't require preconditioning chemotherapy. And he can infuse them back into patients, restoring immune balance, where there's now a nice, better balance between activated T regulatory T cells and activating T cells and regulatory T cells as well. And the goal is that these T cells, when they're infused back into patients, will eventually grow, thrive, live for a long period of time, and control disease activity, maybe drug-free, because now, again, the immune system is balanced as well. So in summary, we spoke about a novel subset of T cells, the T peripheral helper cells that localize in the synovium and provide help with B cell differentiation and immunoglobulin production. B cells, in turn, are key pathogenic drivers of autoimmune response and RA disease activity in patients who have RA. Therapies that more effectively deplete B cells show promise for treating severe and refractory RA. And I can tell you there are lots of bites coming down the pike that are being studied, as well as CAR T therapy for patients with RA. And future RA therapies that enhance immune regulatory checkpoints, including those that can agonize PD-1 or that involve improving the efficacy of regulatory T cells, can offer the potential to restore true immunologic homeostasis and control disease activity from the top of the immunologic cascade, not the bottom. And so with that, I'll turn the talk over to Renuka. Thank you.
Thanks, John. That was amazing. Thank you so much for inviting me to chat with you today, looking at my research on the microbiome and the treatment of rheumatoid arthritis. And we're going to take it a little bit back to basics for the second half of the talk. I have no relevant disclosures. So for those of you who don't think about the microbiome very much, I like to start with a few definitions and some background information. The microbiome, as I define it, consists of the microbes and the genes that they're bringing with them that live in a certain environment. And so today I'm really going to focus on the human gut microbiome, but other investigators look at the skin microbiome, the ocean microbiome, the soil microbiome, because microbes play important roles in the environments in which they reside. And my lab is really interested in understanding the interface between the microbiome and autoimmunity, because our goal is to identify the mechanisms by which the human gut microbiome impacts the treatment response to rheumatologic therapies. And I believe that this information will allow us to advance precision medicine for patients with rheumatic disease. So this schematic encapsulates the types of questions that my lab is interested in looking at. And we're interested in how microbes metabolize the drugs that we give to our patients, perhaps inactivating these drugs before they even get into the circulation. So the catchphrase that I use there is, do bugs act on drugs? But interestingly, we also have come to appreciate that the drugs that we use on our patients, even though they were designed to target our own host pathways, they have off-target effects on the microbiome. And given that I'm going to show you that the microbiome affects many host pathways, it may be that some of the drugs that we currently use have their therapeutic effects by acting on the microbiome and, for example, suppressing immunity or autoimmunity. And then a third question that my lab is interested in is, how does an inflamed host impact the microbiome? And the reason I think this is an important question to answer is because many clinical trials are currently underway trying to use fecal microbiota transplant to treat autoimmunity. But when you transplant these microbiomes into an inflamed host, what happens to those microbiomes? Is the immune system that's activated shaping those microbiota that have been transplanted? And I feel like we have to answer those questions to really leverage microbiome medicine for advancing treatment. So my lab employs multiple orthogonal systems to understand drug microbiota immunity interactions. In particular, we use anaerobic microbiology to understand the direct interactions between drugs and the microbiome. We also use patient samples, and I get a lot of slack for studying poop, but that's what we do. But the clinical samples really allow us to understand the clinical relevance of the observations that we're making from our microbiology experiments. And then we also employ mouse models, in particular notobiotic mouse models. And notobiotics is just a fancy word of saying germ-free. These are animals that don't have a microbiome. You can then transplant them with a specific microbe to understand how microbes influence the host in vivo. And so I'm going to share two stories with you using that illustrate kind of the findings that we've made using these multiple orthogonal systems. Now, I think as physicians in this room, we've all had the experience of prescribing a drug for patients and seeing that the same drug doesn't impact patients in the same way, right? And so this is particularly true in the treatment of rheumatoid arthritis. And John kind of mentioned in his talk that like our primary therapy for first-line therapy for treating rheumatoid arthritis is methotrexate, and that's the drug that I'm going to talk to you about today, how the microbiome influences methotrexate response. So for methotrexate, just like for many other drugs, not every patient responds the same way, and about 1/3 of patients do well, but 2/3 of patients either have a middle or poor response to methotrexate. And for decades, investigators have looked at whether host factors like genetics, environment, diet, the state of the immune system, or other medications might contribute to this variation in treatment response. And what I'm going to show you today is evidence to suggest that the microbiome contributes to this variation. And this is not just true in rheumatology. This contribution of the microbiome to treatment response has been implicated in clinical disciplines as in treatments as diverse as HIV prophylaxis and immunotherapy for cancer. So some of you who don't think about the microbiome, might be wondering, like, is the microbiome important? Like all these host enzymes are probably more important than the microbiome. And so one of the ways that we like to ask whether something's important is to get rid of it, right? So we can do that with these germ-free isolators where we can rear germ-free animals. And what we come to appreciate when we compare germ-free animals with colonized counterparts is that germ-free animals are nutritionally underdeveloped. And that's because we need our microbiome to harvest energy from the foods that we eat and to synthesize essential vitamins for us. Furthermore, the microbiome, the germ-free animals have a stunted immune system compared to their colonized counterparts. And we've come to appreciate that the microbiome educates the immune system and may also contribute to disease pathogenesis. And so it used to be thought like that the immune system was there to keep microbiome in check. It turns out that that conversation goes both ways, that the microbiome can also influence the immune system. And then third, one of the surprising findings is that animals without a microbiome, they have neurologic deficits, including anxiety, hyperactivity, and learning and memory deficits. And so these are just 3 ways in which the microbiome can affect host physiology. There are a number of others. And because of the properties that the microbiome confers to us, we think about the microbiome as extending the capabilities of our own genome. And because of that, they're a second genome to our own genome. And if we understand more about the microbes and the genes that they bring with them, we can leverage this information to advance therapy for patients. Now, there's 2 important facets of the microbiome that I want to kind of highlight for you in appreciating the data I'm going to show you today. First is that the microbiome is a metabolic powerhouse. So the microbiome encodes 150-fold more genes than what's encoded in our own genome. And we need those genes to do important things for us, such as harvest energy from foods and synthesize vitamins. But those same enzymes can act on the drugs that we give to our patients. So known examples of this is like sulfasalazine that we use in rheumatology and gastroenterology is actually converted from a prodrug into an active metabolite. Digoxin, it gets converted to an inactive metabolite by the microbiome. And a drug like irinotecan can actually be made to be more toxic by the microbiome, leading to diarrhea and nausea in patients that are receiving that chemotherapy. A second facet of the microbiome that is important is that it's a potent immunomodulator, as I kind of alluded to briefly in the previous slide. And the mechanisms by which the microbiome can modulate the immune system are actually multitudinous, but a few of them are illustrated here on this slide. First, microbes can produce metabolites that are immunomodulatory, such as short-chain fatty acids, ATPs, indoles, and succinate. And the cool thing about these metabolites is even though they're produced in the gut, they can enter into the circulation and affect distal sites such as the brain and the joints and immune cells at these distal sites. Microbes can also translocate across this epithelial barrier, especially if it's compromised in many autoimmune diseases. And so that's thought to be a leaky gut phenomenon. Microbes also have on their cell surface immunomodulatory metabolites such as LPS. And then a fourth mechanism that I'm not going to talk about too much, but this is one of the sort of ways that the microbiome is thought to contribute to the pathogenesis of autoimmunity is through molecular mimicry or cross-reactivity. So microbes may encode for proteins that have homology to our own enzymes, and then the immune system appropriately generates an immune response, but that can result in friendly fire and autoimmunity. And so a little bit about methotrexate. Even though there are really exciting new therapies underway in the treatment of rheumatoid arthritis, still an anchor drug in RA and a cornerstone drug is methotrexate. As John highlighted, it's the first-line therapy in rheumatoid arthritis. And this is because of many studies showing that it's better tolerated than many disease-modifying anti-rheumatic drugs. It reduces mortality in rheumatoid arthritis. It makes other drugs work better. So you might have noticed that in the rituximab slide that John had shown, that the combination of methotrexate with rituximab was better than either drug alone. And then we use it in a lot of other therapies besides rheumatoid arthritis. It reduces the development of neutralizing antibodies to biologics. So even though it's an old drug that's been around for a long time, I still think it's a really important drug that we want to try to leverage that to get the maximal utility for our patients. Now, for those of you who are interested in history, methotrexate was one of the first rationally designed molecular medicines in the modern medical era, right? So it was originally designed actually to be a cancer therapy. And Joe Bertino was one of the investigators that produced these seminal studies about methotrexate, highlighting it targets the folate pathway. It actually helped us understand more about the folate pathway. And so when he was asked to give a memorial lecture, he decided to give an ode to methotrexate. And I think we also have an ode to methotrexate in rheumatology, given all of these beneficial properties that methotrexate confers. But there's a rub, which is what I illustrated to you earlier, that only a third of patients do well with this compound, with methotrexate, and 2/3 of patients don't. And, you know, as a rheumatology fellow, when I was first starting in the clinic many years ago, I was having this conversation with patients. I would sit down with them and say, you know, I'm sorry you have a diagnosis of rheumatoid arthritis. And, you know, the first line therapy for this is methotrexate, but only 1/3 of you will respond. And it's going to take us 3 to 6 months to figure out if you're going to be a responder or not. And so, of course, that's a challenging conversation to have as a physician and a challenging conversation to hear as a patient. And so one of the questions that I had when I went into the lab was to ask, is this variation that I'm seeing in the clinic due to microbial metabolism of methotrexate? And if it is there a way to nudge people that are in this category into the category of patients that respond well by modulating the microbiome? And so this was an attractive hypothesis to pursue because methotrexate targets this conserved pathway, the folate pathway, which is seen in bacteria, it's seen in plants, it's seen in archaea. So it's all 3 domains of life leverage this pathway for key cellular processes such as purine synthesis, cell cycle, and protein synthesis. And you can see that methotrexate targets this enzyme, dihydrofolate reductase. And the way that it does so is that it mimics the natural substrate of DHFR, which is a folate. And so I'm showing you here the structure for folic acid and methotrexate. You can see they're very similar. All folates have this 3-part tripartite structure with a double ring, a benzoate ring, and then a green glutamate moiety. And methotrexate looks just like folate, except at those 2 sites that are highlighted in blue. All right, and so that's the way it competitively binds and inhibits DHFR. Now, it turns out that we need folic acid in our diet. We can't make it. It's the reason that our grains are fortified with folic acid. It's the reason that we prescribe folic acid to pregnant women. But microbes can make folate, and it's one of the things that they confer to us is the synthesis of folates. And it stood to reason that if microbes can make folate, can they also break down folate? And in doing so, can they also break down methotrexate, which looks a lot like folate? And so it turns out that there was evidence in the literature. If you go all the way back to the 1940s when methotrexate was discovered, these investigators in Bethesda, Maryland had kind of foraged the soil in the creeks around NIH and found soil bacteria that could, you know, use methotrexate as the sole source of energy, but by cleaving this glutamate group and using that as energy source. And they did so with an enzyme called CpG2. And then that led to this compound called DAMPA, which actually is not active at all in inhibiting DHFR. And so here we have an example of a soil microbe that can inactivate a drug that we give to our patients, but that was in soil bacteria. So a natural question was, can this happen with human gut bacteria? And it turns out that nobody had shown that to be true until I did this experiment in the lab where I took individual microbial species and incubated them with methotrexate and then looked using HPLC over time at what happened to methotrexate. So here is this black peak just to the left of the yellow band, and you can see that over time that peak is disappearing and a new peak in yellow is appearing here. And so this strongly suggests that C. asparagiforme can metabolize methotrexate. We then were excited about that result and decided to try 45 different other bacterial species that are commonly found in your gut and my gut. And we found that multiple of them could metabolize methotrexate, as shown here in red. They all come from this Firmicutes phylum, which is this big category that you might have read about in the news because people talk about Bacteroides to Firmicutes ratios. But many bacteria, basically, that we tested could metabolize methotrexate. And then this guy is actually not a Firmicute. That was our positive control. That's the soil bacteria that previously had been shown to metabolize methotrexate. So, of course, being a rheumatologist, I was like, what about RA patient stool samples, right? Do they contain bacteria that can metabolize methotrexate? And so this is where the poop studies come in, where we took stool samples from patients before they started methotrexate, and then just streaked out the samples on Petri dishes, picked colonies that represent bacteria, and then asked how many of these bacteria could metabolize methotrexate. We used sequencing to identify the identities of these microbes, and we actually used a procedure to enrich for the firmicutes. That's why we have so many firmicutes here. But we found, again, that many RA-associated bacterial strains can metabolize methotrexate, as highlighted here in red, showing that there's significant depletion of methotrexate from the culture media. So again, those were 2 in vitro studies, right? Testing one bacteria at a time. A natural question is, what if you have complex microbial communities from RA patients? Is metabolism by RA microbial communities associated with clinical response? That's the kind of money that I'm looking for here, right? And so collaborating with Jose Scher, who had been collecting RA stool samples for a really long time before I even started this research, we also collected stool samples here at UCSF. These are treatment naive individuals. We took their complex microbial communities, incubated them with methotrexate over 48 hours, and looked at what happened. And we can see that some people have microbial communities that quickly metabolize the drug, whereas other people have microbial communities that really don't. And what's cool about our patients is we follow them out after 4 months of treatment and then coded whether they were responders or non-responders. And it's hard to see here, but people who are non-responders tended to have microbial communities that quickly metabolize the drug. And that's shown a little bit better here, where instead of using a yes-no categorical, we kind of looked at DAS28, and people who are out here on the far end of the x-axis do better. People that are over here on the lower end of the x-axis did worse. And if we looked at the ability of their microbiomes to metabolize methotrexate, what we see is that the people who did worse had microbial communities that tended to metabolize the drug. So this is exciting because it supports this idea that microbial metabolism of methotrexate is associated with clinical response. Now, that was in vitro, then we showed this association in patients, but can we actually demonstrate a causal role for the microbiome in vivo? And for this, we then had to turn to our mouse experiments, using that question that I kind of hearkened to earlier. What happens to methotrexate pharmacology if you get rid of the microbiome? Is that change the methotrexate pharmacology in vivo? And so for this, we measured methotrexate, as well as what we think is an exclusively microbial metabolite of methotrexate called DAMPA. We looked at germ-free mice, or mice colonized with a normal microbiome, and then treated these animals with oral methotrexate. And as expected, or excitingly, I think, the mice that are germ-free showed significantly higher peak plasma levels of methotrexate compared to their colonized counterparts. And so this supports the idea that the microbiome contributes to methotrexate pharmacology in vivo. We then also looked at DAMPA, which we think is an exclusively microbial metabolite. And we again see that, germ-free animals don't produce this metabolite, whereas the amount of this compound is increasing steadily over the course of this PK study. And it's only produced in the colonized animal. And what this suggests is that this compound is made in the gut, but it's absorbed into the circulation. And I don't know how much is still there in the gut. There may be more, right? So this may be underestimating the amount of metabolism that's happening in the mice. Now, the levels of the metabolite are 3 nanomolar. Sometimes people are like, well, microbes, do they really play a role? Like, what about host metabolism? Does that play a bigger role? And so we also looked at the host metabolite of methotrexate called 7-hydroxymethyltrexate. A hydroxyl group is added here. And you can see there's comparable levels of the host metabolite versus the microbial metabolite. But what was so cool and so unexpected was that actually there's more of the host metabolite in the colonized mice, right? So this suggests that the microbiome not only influences the production of microbial metabolites, it's also modulating host systems, like the host liver. And so the enzyme that does this in the liver, it's already been shown to be aldehyde oxidase. And that kind of makes sense because the liver is sensing not just methotrexate, but so many other metabolites that are being produced by bacteria. And it turns out like many of our organ systems are awash in microbial metabolites, and we don't really understand what their function is on our organs. Okay, so the previous experiment was kind of like artificial, right? Because we don't have germ-free humans walking around, but we do have humans walking around with different microbiomes, right? And so maybe a closer correlate to what happens in real life is if we have mice with different microbiomes, do they have different methotrexate pharmacology? So does inter-individual variation in RA patient microbiomes contribute to differences in pharmacology? And so for that, again, we started with these germ-free mice. We colonized them with stool from 2 different RA donors. We were selecting these samples to be very different in terms of their microbiome composition and treated with oral methotrexate. And what you can see here is that, mice that got this patient's microbiome had significantly higher levels of methotrexate than mice that were colonized with this. So this, again, supports this idea that variation in our microbiome can influence drug pharmacology in vivo. So in this first part of the talk, I showed you that human gut bacteria directly metabolize methotrexate in vitro, that this metabolism is associated with clinical response, and that in vivo, we see that the microbiome modulates drug pharmacology. And so a very simplified schematic is using these 3 model systems. I provided evidence to support like this relationship between the microbiome and methotrexate. But many questions remain unknown, including, what are the microbial genes involved? This is a question that I get a lot from patients, can we modify the microbiome through diet and affect this metabolic activity? And obviously we want to answer these questions because there are clear clinical implications to understanding how to modulate the microbiome. If we know the genes that are implicated, maybe we can use CRISPR to edit out specifically those microbial genes. So again, lots to learn. Okay, in the last 5 minutes, I'm just going to go, I showed you evidence for this first direction. You never know where science is going to take you. I started this project looking at metabolism. One of the things I uncovered was that when I put methotrexate with bacteria, the methotrexate actually inhibits the growth of some microbes. And so is methotrexate reducing inflammation by modulating the microbiome and reducing its inflammatory potential, maybe by reducing like the production of proteins that confer this molecular mimicry and cross-reactivity, right, that lead to rheumatoid arthritis. And so it had actually been known a long time ago that methotrexate can bind to bacterial DHFR, but bacteria that were most studied at the time, like E. coli, shuttle that drug out, just like they shuttle antibiotics out. And so, many of the bacteria that had been studied had been resistant to methotrexate, but people hadn't really had the opportunity to look at human gut bacteria. And so that's what I did here, where I took C. asparagiforme. This is just by itself in culture, incubated it with 10 different concentrations of methotrexate and looked at growth. And you can see C. asparagiforme is pretty resistant. Maybe this highest concentration causes a little bit of a decrease, but not really, right? And that's in contrast to Bacteroides theta iota omicron, where increasing amounts of methotrexate can completely inhibit the growth of B theta. And so this right here, 112, is the minimum inhibitory concentration, or the MIC. And you guys use this measurement all the time whenever you send cultures. What you're getting back is an MIC, right? So that's the minimum inhibitory concentration. And so here is another more sensitive Bacteroides fine-Goldii. So lower MICs imply that the bacteria are more sensitive, okay? So again, we went back to our panel of 45 different isolates and looked at their MICs. And you can see that as a heat map, there are some bacteria that are very resistant. Those are the dark red, and then others that are very sensitive in the light. When we kind of stratify these bacteria based on those major phyla, the big categories that encapsulate many species, we see that Bacteroides tend to be sensitive, okay? So this is in vitro, the Bacteroides tend to be sensitive. We wanted to know in vivo, and so we turned to mice that were colonized with a healthy human donor. And what we see when we treat the mice with either saline low dose or high dose is, again, this Bacteroides phylum is coming up as being inhibited by methotrexate. And then finally, we looked in patients and saw the same thing. So we took patients' stool samples before methotrexate and 1 month after methotrexate. And it's kind of a mixed bag when you look at all patients together, but when you stratify them by responders and non-responders, we see almost every responder to methotrexate showed a decrease in, again, that same phylum, Bacteroides, right? So this decrease in Bacteroides that's kind of induced or associated with methotrexate is associated with clinical response. And so that's what gave us the motivation to then test in vivo. If you take mice and you transplant them with microbiomes or microbiota before the patient gets methotrexate or after they get methotrexate, is the microbiome from the patient after they got methotrexate less inflammatory, right? And that's exactly what we saw, that mice that had microbiomes from the post-methotrexate or methotrexate-exposed microbiomes had reduced immune cell populations of many of these immune cells that are implicated in rheumatoid arthritis. And so we looked at the spleen, the small intestine, and the colon. And so that was exciting because it suggests that methotrexate not only affects the host, but also affects the microbiome to reduce inflammation. So in part 2 of this talk, what I showed you is that methotrexate alters community composition in mice and in RA patients, that fecal microbiota transplant with methotrexate exposed communities led to reduced immune activation. And I didn't have time to show you this, but antibiotics in a mouse model can also alleviate arthritis. And so, if there's, right now, we're doing a lot of things to target the immune system, but we need to go all the way upstream, right, to maybe the bacteria that are inciting that immune response or triggering those flares, and target the microbes and have microbiota targeted therapies to reduce inflammation. And so again, there are many questions that remain unknown. and have strong clinical implications. Now, the last thing I'm going to share with you in the minute that I have is that can we use information from the microbiome to predict response? Like if the microbiome metabolizes the drug and is also a target of the drug, can we identify patients when they walk into the clinic and when I give them that diagnosis about whether they're going to be a responder or a non-responder? And so for this, we did shotgun sequencing of treatment-naive patients of their microbiomes and found that the microbiomes of people that go on to become responders is significantly different than the microbiomes of people that go on to become non-responders. And we can build a machine learning model to predict response that outperforms clinical and existing pharmacogenetic models. And so this was done in an exploratory cohort and a validation cohort, and now we're doing it again in a third validation cohort that spans multiple institutions. And so in summary, I do think that genetics and social determinants influence the response to methotrexate, but the microbiome does as well. And this is one of the components that we can modulate. And so, the model that I'm operating under at this time is that people who are responders, they tend to have microbial communities that are sensitive and may also contribute to the aberrant immune activation. And so methotrexate targets both the microbiome and the immune system to alleviate RA, whereas patients that go on to become non-responders may have microbes that are resistant, that may be metabolizing the drug and preventing methotrexate from doing its thing either on the host or on the microbiome and alleviating RA. And so with that, my take-home messages for you guys is that microbiome represents an underappreciated source of variation in drug response, that we can leverage the microbiome to advance precision medicine for patients with rheumatic disease, and that if we understand the pathways that are implicated, we can develop microbiota-targeted therapies to advance care. And with that, I thank you for your attention, and I thank the people that helped carry out this work and our collaborators and our funding sources.
While you all are thinking up your question, I'm going to ask one of mine for Dr. Nayak. The world of drugs out there that you might consider this for is vast. Methotrexate seems like a great target, both because we've known so much about it, we know the one-third, and because it makes sense physiologically with what we know is happening in our gut microbiome with metabolism. How do you go about thinking about other targets for this? Have you started thinking that is this applicable to other drugs in cardiovascular health or, antibiotic, whatever the alternative drug? Yeah, I'm curious.
Yeah, so there were two seminal studies, one where they did a screen of many drugs and tested in vitro whether microbiomes from different people or maybe different bacteria can metabolize like a panel of 1,000 drugs. And so in that screen, they found significant, like 200 drugs that were metabolized by microbes. And these are drugs that we use, in modern day medicine. Now, what the clinical implications of that metabolism were not evaluated in that screen. And so it really, I think, depends on physician scientists like us who are interested in understanding, well, what are these metabolic transformations? What is the effect on the host? And so there's evidence that many drugs are metabolized by microbes, but again, we don't understand what the clinical relevance of that metabolism is. And the other kind of question has been asked too, that how many of the drugs that we use currently, they again did a screen and found many non-antibiotic drugs can influence, again, they did kind of a panel of 45 different bacteria, can influence the growth of these bacteria. And again, that was in vitro. So what happens in vivo is also not known. And so really we need physician scientists to answer questions like that are kind of relevant to our patients.
Questions from the group? Yeah, Alan.
Yeah, I've got a question. You said patients would want to know if there's anything they could eat that could help. Do you see any role for probiotics? I get that question from patients all the time. Is this a role where probiotics might actually have a benefit?
So I think that there's a lot of studies that look at probiotics. It's hard to interpret them because and like with drug formulations where there's like a standard production of them, different probiotics have different formulations, right? And so there's definitely, if you look in rheumatoid arthritis, there are some small studies of probiotics benefiting patients with rheumatoid arthritis. And I think you can find correlates of that in other diseases. I think that there is now this next generation of live biotherapeutics or LBPs where people are looking at, strains that are normally found in the gut. Can you engineer them so that they produce metabolites or other kinds of, can they shift the microbiome in certain ways? And so the short answer to your question is that I don't know of very many probiotics that I could recommend right now to my patients. There are some like VSL3 that existingly right now physicians are prescribing to their patients. but I still think that this is an emerging area, and there's new techniques that are kind of being used to genetically manipulate, Akkermansia and other strains that are not classically considered as probiotics that are being considered now. Yeah.
Just a correlate to that, how about certain foods, not supplements, but foods, fermented foods, etc.?
Yeah, I think foods is such an interesting question, because if everyone's microbiome is different, then there's evidence for this too, that the way that they respond to certain foods will depend on their microbiome. So now there's this evolving field of not just precision medicine, but precision nutrition. And that, maybe the diet that I recommend for one person may not work for a different person. And again, there's people that are kind of looking at that, but then you need these kind of like multi-omic strategies in humans, right? To document how is everyone's microbiome different, and how does that influence how diet shapes the microbiome and then shapes the immune system. Yeah, I think in our study, there are some patients where I think the microbiome plays a role in the pathogenesis of RA. And so in those patients, maybe targeting the microbiome RA we call it RA, but it may be many different diseases that have different kind of triggers in the lung or in the gut or genetics, right? And so again, diet, it's hard to then figure out like how to guide patients on diet. One thing is very clear, that we kind of recommend people have a healthy diet. And so if you ask 10 different rheumatologists, one will be like Wales anti-inflammatory diet, another person will be Mediterranean diet. And so that to me suggests like we still need to do a little bit more work there. Yeah.
Unfortunately, we need this room. So I will thank our two excellent speakers who did just an amazing job. Thank you to both. It's terrific.